everybody. Do you need to stretch? No stretches? Okay, great. I think probably a lot of people are waiting to hear this talk. And I know from my patients who are visiting stem cell clinics that they are anxiously awaiting to get some science behind the snake oil. So am I. So I hope that I will be able to enlighten you. I'm going to spend a bit of time in the slide deck on biotourism. I want you to understand what's really going on in a field that it's really in its infancy. So if you look at this, this is in recent years the number of clinical trials involving stem cell therapy has increased worldwide. Now, when we talk about these clinical trials, they're not necessarily real clinical trials, because as Lee Huff has shown in a very interesting paper on clinicaltrials.gov, a lot of what's on clinicaltrials.gov are not really clinical trials. The independent stem cell clinics have figured out a way to get themselves on there. They masquerade as studies. They even use consent forms, which are truly bogus. And so when I show you this, keep in mind what you're looking at. But just look at, I think the real emphasis here is that this is a global problem. This is not just the United States. But North America is leading. And probably where I come from, Florida, is one of the leading sites. But if Lisa Lancaster's here, I know Wendy's here, just down the street here from Vanderbilt is one of the big stem cell institutes, quote unquote, where we charge $14,000 a treatment. So these are the active clinical trials by cell type, and what I just want to show you here in my brief moments of talking about this is that you'll notice that the lung is missing on this side. There have now been many more trials. In fact, when I did this last week, I got 5,040 hits under mesenchymal stem cells, and last year 493. So it's on the rise. There are trials in asthma, there are trials in bronchiectasis, there's trials in COPD, even though we've published on COPD. Dan Weiss did that. We've got the ETHER trial. It's a growing number. So what are the facts? The 21st Century Cures Act of 2016, which was Public Law 114-255, created the designation of regenerative advanced therapy. This followed the ReGrow Act, which I know Sonia has been very involved with the FDA in understanding this. And the real intention here was to accelerate approval of stem cell-based therapies and bypass stringent testing required by the FDA. Part of the problem here is this minimal manipulative category that allows these clinics to process your cells in a day and give them to you without any regulation. They are minimally manipulated. However, you're going to have a liposuction, you're going to have some kind of spinning procedure, and you're going to have stuff shot back into your arm. Who knows what that is? I don't know. It's mush, if you ask me. Commercial stem cell bypass, these clinics bypass the regulations, as I've mentioned, and they offer cell therapies for a dizzying list. I quote George Daly from Harvard, a dizzying list of diseases from autism to heart failure. Anything you got wrong with you, we got a cell for you. They market directly to the patient through the internet. It's called direct-to-consumer marketing. Goes right to the patient. The patient then replies online and gets immediate feedback with a chat function so that they too can be tied into the clinic as soon as possible. Here's the real problem. The last bullet. Patients have died. 3 others have recently been blinded. The blinded patients, as you saw in the New York Times, it was published in the New England Journal, both eyes were injected at the same time. They were then brought to Bascom Palmer at the University of Miami because unfortunately this was a clinic in Fort Lauderdale. Those women have remained blind, and this was the only thing, finally, even though they had had deaths at this site in Fort Lauderdale, that actually brought the FDA to shut that place down temporarily. It's not shut down permanently in Sawgrass Mills in Fort Lauderdale. So what's in a name? What's stem cell? What is this? So we have embryonic stem cells. They're from the inner cell mass of blastocysts. We have induced pluripotent stem cells. These are adult cells that are reprogrammed for pluripotency. Take fibroblasts, put them with a retrovirus. We can get iPS cells. We can get these cells to do lots of things, but they're virally transformed, so to speak. You can make them from mice, first reported in 2006. awarded a Nobel Prize for Human, 2007. And then we have the mesenchymal stem cells and the hematopoietic stem cells that are made from your bone marrow. So we have a lot of cells in our body, as we all know, that can go through different tracks to do different things. What we're going to focus on is these mesenchymal stem cells, because the hematopoietic ones are the ones that are subject of when patients with leukemia, for example, get bone marrow transplant. Those cells are coming from the hematopoietic stem cell line. And then, of course, how we apply them to the lung. What is stemness? What does that mean when we talk about a stem cell? There are two main properties you need to remember when we talk about stem cells. We talk about the cell that has the ability to self-renewal and the capacity to differentiate into mature cell types. So given enough time, they are engineered to be a definitive cell at the end. So whether that's going to be a cartilage cell, a fat cell, a bone cell, that's how it's all going to be engineered. The second one is they have plasticity, the ability to differentiate into specific mature lineages. This is the most important property that we're using for cell-based therapy. I just show this not to belabor how long it takes, but I want you to understand when a stem cell clinic is giving a patient a cell, this is not a cell that has been processed. They're getting something from the peripheral blood and giving it back, or they're getting it from the fat. They're giving them fat cells mixed with what's called stromal vascular fraction that may have a whole host of cells in it. This is actually part of the process to make a stem cell. And when we get them from the bone marrow, we get about 90 to 120 cc's of marrow. It takes 4 to 6 weeks. This is a bunch of flow studies showing you some of the markers that are required to be able to identify these cells as mesenchymal stem cells. We look for markers that are negative as well as positive. We look for it in serial cultures. These cultures have to be checked for immunogenicity, for microbial disease before, of course, they're given, and they have to go through a special facility called a CGMP facility. So the expression pattern is what we use. It's depicted here with these representative histograms and the expression of very specific markers. The one that we really want to make sure of is that they don't express CD45. So there's actually is some characteristics to these cells now that we can use through different donor populations to be able to identify what they are. This is just a sort of diagram for you how we look at this in terms of cell differentiation. I mentioned to you that these MSCs, the mesenchymal stem cells, can really develop into a whole host of cells. These are cells that can be used for cell transplantation. Once again, we're not talking about bone marrow transplantation because these are mesenchymal stem cells. We also can take these cells, we can see them from the iPS cells that I showed you, and also get them through some reprogramming techniques, right, to do that. And then we can use them in a 3D model to actually construct tissues and to use for tissue transplantation. This is really a very active area now as we use them in an organotypic type of culture system to try to make sort of a mock organ. What's the differences between an MSC and an iPS cell? I'm always asked this, so I put this in a little figure for you. These are MSCs that are fibroblastic-like, and when you look at them in the, under the microscope, versus an embryonic stem cell-like, a much more immature cell. We talked about these markers. We talked about the CD45 negative. You can see that there's a big difference between the markers, so it's not hard to do. You have to take the time, do the characterization. Plasticity, multipotent versus pluripotent. Differentiation, they come mesodermal tissues versus others. This is a much more broader spectrum. Tumorigenesis has not been reported with mesenchymal stem cells, but it has been reported with iPS cells, which is a concern for us, right? As we think something that we're going to take to patients, we don't want to make something that can be transformed into a malignant cell. Definitions, just to review again, I think this is probably the most important piece that I take away from this, is that you know what we're talking about, because I think the language is difficult. Embryonic cells have the capacity for self-renewal, pluripotent, ability to differentiate into all 3 germ layers and all adult types, versus the iPS, where we talk about formation of tissues from all 3 germ layers, and adult stem cells, cells from adult tissues such as bone marrow or adipose tissue, capacity for long-term self-renewal and differentiation, and then last but not least, the progenitor cell. So that's a cell that actually even comes before the stem cell. There's been quite a lot of work done on progenitor cells, not so much in terms of clinical trials, although there are several now with endothelial cell progenitors looking at vascular disease. I won't spend too much time on that because I think the terms start to just become baba ganoush here, and you don't know what's what when I start talking about it. So the current active cell therapy clinical trial showing the breakdown of hematopoietic and mesenchymal stem cells so that you understand what's going on. Here's the hematopoietic cells and here's the mesenchymal stem cells. The majority of clinical trials are based on bone marrow derived cells. So why bone marrow? This is a very simple little characteristic here of why bone marrow. I want you to take from this is that look at all they can do. So if we start to talk about a drug therapy, usually we're talking about a specific pathway that we're going to target, right? Drug companies make drugs to go down and antagonize growth factors that are tyrosine kinase or their G protein coupled receptors. This is not what we talk about in the MSC world. We're really talking about a much more global attack, an attack on innate immunity, adaptive immunity, really changing the whole course of the milieu that the cell is seeing. I love this slide from Viva Lama from Michigan, and it's a gift from her. But I think she really did a great job at really depicting all of the different ways, right, even with NK cells, T cells, and looking at how many attack pathways these cells have. These cells can really make a difference if we can figure out how to make them a homogeneous population, if we can figure out the donors and how to make what we get from the donors work the same in every patient. We are far, far from that. So the stem cell clinics work with mostly autologous cells. So you go into the clinic, they do an aspirate from their abdomen, and then they prepare cells. They're your own cells. So you're a 60-year-old ex-smoker, and you've got lung disease. They're making cells for you, stem cells from you, that I would argue probably haven't worked. But you're going to get your own cells back. The work that we've done is with allogeneic donors. That means that somebody, just like a bone marrow transplant donor would have, someone comes in, gets a bone marrow aspirate, a healthy donor who's been totally screened for HIV, CMV, everything to make sure they are a healthy, clean donor that wouldn't transmit any disease. And then the cells are made from that donor. They're not made from you. You are not your own donor. So that's the difference between allogeneic and autologous. And if you look at what's going on in the field, and you notice this nice little hash line, what's really going on is that in the clinics, right, and out in the world, a lot of people are still working with autologous cells. Why is that? Because it is easier, right? You're your own donor. But one thing you need to know about MSCs is that they don't create, you're not going to see them as foreign, because mesenchymal stem cells lack MHC class TIS. So they don't have the histocompatibility antigens to be recognized as foreign. So this whole idea that you can't use a donor because they'll be foreign and you have to use your own is bogus. It doesn't mean anything. So when you're being told that you have to use your own, it's only for convenience and only because it's faster and only because they can get around the rules that they know about having something minimally manipulated. The lawyers have been very good about this. They can minimally manipulate your cells and give them back to you. And maybe they're going to get you one stem cell, and I don't even know if that cell's going to work. But that's what's going on. And this is put together by Laertes Economo from Boston University and Daryl Cotton's group. And I think it goes up to 2012. He's got some newer data that basically shows the same kind of pathway. So how should this be being done, right? I think this is the most important question that we're facing is how should this be done? So this is the mechanism of clinical translation when we want to develop something. We get an IND, that's really at a phase 0. We always do a phase 1 safety trial. We go to phase 2 in a larger group of patients to try to get a signal of efficacy, and then we do a definitive phase 3, which may or not have a run-in phase for safety, depending if we're using a different source for the material that we're testing, and then looking at efficacy and establishing dosing. What the FDA wants to do, Scott Gottlieb, is to create fast-track breakthrough therapy designations that allow accelerated approval and priority review. And unfortunately, as Sonia's probably looking at this and her heart's in her throat, this piece right here is where the problem comes. So there should be some probably compassionate use. But if we use clinical vignettes to drive this, to allow us just to bypass this whole testing, randomized control, having controls that don't get the cells and seeing what happens to them, we're never going to know if there's an effect. I see lots of patients in the audience. And you say, why do scientists want to do all that? What's this problem? Why can't they just give it to people? You know, somebody, my friend down the street said it made her better. Because we have to test it. We have to know that there's not just some dummy effect behind it, and then we have to know how to measure the effect. And so this is really the process here. The mechanisms I'm going to show you here are that, let me see if I can get this to do it. You know, we have to have really important rationales and preclinical evidence. We have to have peer review. We have to have medical innovation with very serious patients that we certainly target. Clinical trials when possible and proven therapy. But the real problem, I think, is this little track here, which is no rationale or preclinical evidence of efficacy, no peer review. Stop, it looks good, let's just give it to people. And then we have the 3 women who died because both of their eyes were injected. So I would caution patients and physicians. I am often disturbed by the number of physicians that refer their patients to stem cell clinics because they feel that there's nothing else to be done, so why not send the patient to the clinic? I would say be very, very careful of this. Stem cell medical tourism. So stem cell clinics treat a variety of conditions using the same cell type. A lot of these, we've just had patients go to Mexico, Panama, as I've said, and as George Daly has said, this is a dizzying array of conditions. I love the terminology. And this is just a short list, and of course our pulmonary diseases are on here. This is from Laertes, and so this is just the diagram for you of really collecting the fat the same day, putting it through some gobbledygook machine, and then giving it to you. The cell sources tend to be mostly, there are some clinics actually doing bone marrow in Colorado. We have patients that are getting bone marrow aspirates and then injecting in their knees, the soup that supposedly works. They're being charged anywhere from $7,000 to $10,000 for their knees. Adipose tissue we've talked a lot about now. Umbilical cord. So in Mexico we have umbilical cord cells being used. The identification of the background of the mother isn't known. It isn't known if it's a male or female baby. There are several parts of the umbilical cord for you that know this, and we don't know if they're using Wharton jelly or what they're using. They just put some stuff together and then they give it to them. This has sort of come out of vogue right now, fetal. I spoke at the Vatican a couple of years ago, and the Pope was really interested in finding out non-embryonic uses, right, of stem cells, so that he could be able to tell people what might be an approach to this field, because clearly there are some religious issues with it in terms of using embryonic cells. And then there's this latest one that's become very, very interesting, which is the peripheral blood transfusion. So we take the blood out, and then we go to another room, we spin it down, then we come back and we give it to you. And supposedly we have stem cells. I don't think so. So the mode of administration often is intravenous. Sometimes it's intrathecal for some of the neuro diseases. Intramuscular has been used. And the latest one this week I got was aerosolized. Had a patient go to Mexico who came in to see me because of progression of disease. They received aerosolized adipose something or other, and were told that it was experimental and hoped that it would work. So they got aerosolized cells. I think all of us in the audience of the physician group would say they probably just got cell parts that who knows what, if anything, even went in and where it would go. We know from an aerosolized, even medications, we get probably less than 18% down in the lungs. So I'm not sure what they did. So this is sort of a scheme so you understand how it works. And then the business offering stem cell treatment. So this is the direct-to-consumer marketing story. Misleading advertisements. Direct-to-consumer patient testimonies. If you go on any of these websites, Lung Institute of Florida is my favorite. They have movies of these patients where you can hear how they've been cured, but they're sitting there clearly that's not the case, but they feel that they're better. And I already mentioned to you, watch out for clinicaltrials.gov. There are major efforts to clean it up now, and it is in place, the cleanup. So this, I think, will be, the government's taken a stance on this fairly recently. Misrepresentation of risks and benefits, portrayal of treatments as routine. In other words, we do this all the time. We've been doing this for 5 years. We haven't had any problems. What's the difference for you? Exaggerated claims of safety and efficacy. They don't really have any claims. I mean, they don't really collect the data. They certainly don't have any efficacy data. I should warn you, though, that they sometimes have publications. So they manage to get these things published online that actually can be tracked. They're not part of our NIH group, which is the PubMed way, but you can find them on Google search. So patients will think that because they can find an article on this treatment, that it's been validated and it's efficacious. No, it's not. There actually have been no efficacy studies yet that have been shown, that are published, and credible and peer-reviewed. Weak or absent scientific rationale. Absolutely. This is a real problem. The scientific literature appearing in websites is outdated or not relevant. Poor patient follow-up. They don't even know what happens to half these patients. I had a patient pay $100,000 to go to Mexico to Celtec for 10 treatments. There's no tracking in between. They're given reminder notices to come back to Mexico, but as far as following any pulmonary parameters or anything, nothing's done. Absence of regulatory oversight and objective clinical evidence for claim benefits. Clearly, that's one of our biggest problems. The stem cell clinics and FDA regulation. Human cells and tissue-based products are considered drugs. Exceptions to this rule are cell products that are minimally manipulated, intended for homologous use and not combined with other articles, destined for use in the same individual within the same surgical procedure. And most stem cell outfits in the U.S. claim these two exemptions to avoid having their products or treatments considered as drugs. So these are two of the references for that. I've included at the end a whole list of references. But you can see that the claiming of exemptions, that's also not really valid at all. Status of cell therapies for respiratory disease. So this was put together, Laird's put this together for our ATS stem cell symposium that we had. And this just shows you the number of organizations, including the Pulmonary Fibrosis Foundation, that have come out with statements to try to warn patients for this. The Cystic Fibrosis Foundation. This is really, we're trying to get patients aware that it's not that we're trying to stop getting the treatments, we're trying to get science behind the treatments. We want to make sure things are safe and efficacious for patients. This is selected clinical trials of stem cell-based therapies for pulmonary disease. This is from a paper from Lowenthal and Sugarman and Chess. This just shows you ARDS. This whole group has really been led by Mike Mathey at UCSF. Bronchiolitis obliterans, we have a phase 1 Mayo Clinic. Bronchopulmonary dysplasia, this is a therapy known as pneumostem from a private company. COPD, this was Prochymal done by Osiris that's been bought now by a company Mesoblast, but we haven't been able to put another trial together. There's a lot of interest actually in revisiting COPD. Both from better characterizing the patients that could be studied and really looking at it. I think Dan Weiss did a marvelous job at safety, but unfortunately couldn't get an efficacy signal. And here's some other trials. Here's COPD emphysema, adipose-derived, bone marrow cells. Sometimes you get known, they try to label the cell. The thing about stem cells is you do need to know where they come from, but the characteristics that I showed you should be present. So whether you get a stem cell from a bone marrow or you get it from adipose tissue, the markers should be the same. You should be able to characterize them. PAH, both familial and idiopathic. And now included on the next rendition of this will be trials in bronchiolitis obliterans, trials in asthma that we're doing. We've published on ether in patients with idiopathic pulmonary fibrosis, and we now have approval for RECELL that I'll talk about in a few minutes. So the results of human studies of human mesenchymal stem cell therapy. This is just now focusing on the lung disease and giving credit to everybody who's done the work. Dan Weiss, Slakis, Dan Chambers, Wilson. This is Mike Mathey. This is the ARDS, and then this is our paper on IPF. What I point out about this, since this is a group for idiopathic pulmonary fibrosis primarily, is that this is a 3-cohort study. I'm going to show you about this. We use 3 different dosages. Please note the small number of patients that were tested. Anybody who's ever heard me talk about this, I always say 9 patients. Beware. It's a very small study. Don't make any big conclusions, except to say that we didn't have any treatment emergent serious adverse events. We did have 2 deaths outside of the study due to progression of disease, and then the exploratory results I'm going to go through in the next few slides. So let's go back and think about why would we want to be interested in doing this in IPF? What would be the story here? And this is just from the original paper that we put out on the perspective, which was 2013. We subsequently got the first approval from the FDA. But the idea is that these cells could really have, as I've shown you, really multiple mechanisms of action. So if we're thinking about a disease that's as complex as IPF, and we're thinking about more of an oncology paradigm, where we have to use many drugs to treat the disease, how about if we could use one therapy that it would attack many different parts of the disease? That's what stem cells could potentially do, right? Because of all the things I've showed you about what their capacity is. And so that's what we used when we went to the FDA, that we thought that they could both be important in the inflammatory phase that we didn't really get to see clinically, but also in maybe some of the structural changes that we saw in the lungs. I want to give credit, though, to all the people who did the animal work, and credit to the minds that went behind this before we took this to patients, and recognize that we don't really have a true animal model of IPF, except that we've now published the model along with a couple of other groups and really looked at older male mice, since the majority of patients who get IPF are men above the age of 50 who are ex-smokers, and we've taken that model to old mice, and that the Bleo model actually, through several papers now, shows a lot of overlap with patients with human material from patients with idiopathic pulmonary fibrosis. And this is just to show you the model. This is a mouse that's given intratracheal bleomycin. This is day 3. This is day 7 with the blue color showing you the fibrotic areas with collagen being laid down, and really a very diffuse process by day 14. Credit to Luis Ortiz, who in 2003 was the first one to give these mice mesenchymal stem cells. You look now at control, you look at the bleomycin, which is the same as the day 14, and the bleomycin with the MSC is looking much better. Luis has a long-standing interest in this area. He's at the University of Pittsburgh. Where do they go? So this is the other piece that's a little challenging, is that in most of the studies, it appears that the cells go to the lung, but they don't stay there. They sort of drop off their contents, whatever that would be, and then they journey on. So this is a study in dogs, with the dog lying on its side. It's actually a cardiology study using these oxine-labeled MSCs. Within 1 hour, it's in the lung, and within 24 hours, it's in the liver. Now, clearly, we can't do these studies all in humans, right? Because we're not going to biopsy them to find that out. But we have an ongoing study now with labeled cells in animals to look at this. And there's been one labeled study in humans in Greece that has actually looked at where the cells might go. Much of the data is reproducing itself in the sense that it goes to the lung very quickly, dumps on, moves on through what's called the reticuloendothelial cell system. This is just, I don't want to spend too much time on this, the preclinical animal studies to really show you that these studies for a long time, I mean, if you look at the years that were put into this, really credit to all the minds that were behind this, no adverse effects, they looked like there was some efficacy signals. These were all the animal studies. And then what really struck us was the story that in fact, all of the animal studies that I showed you in that slide were done in young mice. Let me remind you again, who gets IPF? I don't see it in children. I don't see it in young adults. I see it in men over the age of 50 who are ex-smokers. And so we took that model to old male mice. We did a couple of things with this model. Ivan Rosas and others, Mauricio Rojas actually, did it too with Anna Mora, looked at old animals. We did this and showed that here's the saline animal, right, at a low and high power, and the animal given bleomycin, right, with young ASCs. So what we did was the donors were either young or old mice, their bone marrow, and then we gave them to animals who had the fibrosis, who had developed bleomycin fibrosis. This is a preventative model, right? So we did both giving the bleomycin and the cells on day 1, that would be protective. That doesn't really translate to humans, right? Because then I'd have to screen populations, I'd have to assume that so many people would be getting fibrosis, and then I'd be giving them treatment. And so here, we gave them old ASCs. You see they don't work. So the old cells from the old mouse do not do anything to the disease, but the young cells seem to really repair this disease. So let me go quickly through the trial. Mary's pointing out, this is intravenous, this is published now in CHEST last year, intravenous human MSCs, demonstrate safety, please note safety, with exploratory efficacy, looking at lung function, acute exacerbation, change in symptom and survival. It's a phase 1. I mentioned the 3 cohorts. It's intravenous. They were followed for 60 weeks. They've now been followed for even longer. 9-patient pilot safety. And to show you the primary endpoint, we're looking for treatment-related adverse events, and the secondary endpoints were here. Let me just go quickly through this. This is just the demographics of the patients. Any of you are interested in this, I can go through this in more detail later. What did we find out? So we used a fairly homogeneous group of patients, men about the same age, about the same level of the disease, and what we found out was that there were no treatment-related adverse events. The most common adverse event was bronchitis, which has been shown in other IPF trials. And the selected parameters that are associated with increased risk of mortality in all of the studies that we've looked at have been to look at effects in FVC, 6-minute walk test distance, which isn't too great, and diffusion capacity. What we were able to show was that in these patients, if you notice, there's quite a bit of what you call straight lines. They seem to have quite a bit of stability when we treated them in terms of their absolute FVC declines. This is looking at just 4 of the patients in cohorts 2 and 3, the more advanced patients, and then looking at 6-minute walk distance. And when you start to see this, right, this data, you say, gee whiz, and all the studies that all of us have been involved in, and if we think about what kind of improvements we would look in 6-minute walk distance, this really looks pretty stable. But the one piece of data that really led us now to do the multi-dose study is actually the diffusion capacity data. Let me show you. Where we saw that these patients had, in all the cohorts, really this very interesting stability. But what happened at 6 months, if you go back, right, and you look at some of the other data, was that we could tell that they were starting to decline at about that point at 6 months. Makes sense, right? Would we really think that one dose of this would ever do anything? And remember that it's totally exploratory efficacy, right? You only have 9 patients to work with, so it's a lot of dreaming. But it was the dreaming that led us to understand that maybe a multi-dose trial was the way to go. That's what we've recently been approved for. Patients will get dosed every 4 months. They are getting cells from the same donor on each dose. It's a multi-site study where we'll finally get some data behind this to really standardize what patients are getting. So the mean change was really below 5% as compared to a greater than 5% decline that's been reported in all the other studies. The DLCO, remember this is just a dream, improved at 24 weeks. We looked at the 6-minute walk test. We looked at the FVC. We started to realize that we needed to do more of this. And the DLCO data was the strongest data we had, which is 7 out of the 9 patients to support 100 million dose, which is a very sort of superficial way to decide on this, but that's what they're going to get. They're going to get 100 million cells. And the most important thing, I think, is that we can be assured of the safety in many trials. Wherever we've looked at all of the reputable trials in cardiology and neurology, the cells, I don't think we have safety concern. We have more concerns about what actually can be standardized to be given to the patient reproducibly, like a drug. That's where we have to go. So what direction are we headed? Anecdotal reports have superseded hard evidence, and patients are victims of an industry peddling unproven and experimental stem cell-based treatments. I don't want my patients to be victims, and I'm sure you all don't want to be victims. We need rigorous, double-blind, randomized trials. We need to put real science behind it and keep doing it. Push the pendulum because we need it. Safety is established, but not efficacy. I think we're very optimistic RECELL will be the first multi-dose randomized controlled trial in patients in IPF, and I've said many times at this meeting already, if the study is negative, it's as valuable as if it's as positive, because we'll get the information out to the audience that needs to know, our patients and our physicians. This is just the latest and the greatest on how many patients are really at risk, and so I'll be happy to entertain any questions. This is a list for you of some of the resources I used to prepare this. Thank you very much.
What an amazing talk. I'm so optimistic about the future and all the good work that you are doing. So excited. Does anyone have any questions about stem cell transplant? When it's done correctly, does it give you side effects? Are there side effects associated when it's done correctly?
No, so patients don't really experience too much. These cells don't really elicit any immunogenicity at all. And as far as volume, there's been some concerns about the clinics out in the community about how much volume is being given. Some patients have gone into failure. We've had patients die from fat emboli. I guess Sonia and I get the privilege of seeing some of these cases for review, and it's really heart-wrenching. But truly, it's fairly safe. They don't really have any major side effects, no.
Thank you so very much.